Thank you for the introduction, and thanks to the organizer to give me the opportunity to talk today. I'm really pleased to be here and to present actually the two therapeutic approaches we are implementing in the lab in order to try to rescue CMT2A. So, as you know, CMT2A is the most frequent axonal CMT, and it is often associated with an early onset and severe motor neuropathy. It can be also associated with sensory alteration and in few cases to optic atrophy. So CMT2A is caused by mutation in the MFN2 gene, and up to now more than 100 mutations have been described so far, mostly with autosomal dominant transmission all along the gene, despite some hotspot region that make also with all these mutations the therapeutical development difficult. And so MFN2 gene encode mitophysin 2 protein, which is a GTPase protein that is anchored in organelle membrane and that act through either homodimers or heterodimer with MFN1 thanks to the hepta repeat domain. And initially, so MFN2 is well known to be located at the outer membrane of mitochondria, where it regulates mitochondrial fusion. But during the past 15 years, more groups have shown that MFN2 is also located at the ER surface and help to the interaction between the ER and the mitochondria. And these contacts are really important for this organelle function, as well as cell function and neurons, because they regulate calcium homeostasis, lipid exchange, autophagy, also mitochondrial dynamics. And in our initial study published in 2019, we have shown that the most common mutation in MFN2, which is the arginine 94 glutamine, led to a disconnection from the ER and the mitochondria that we observe both in the motor neuron of the spinal cord of our CMT2A model, which is a MitoShark model, as well as, thanks to collaboration with Vincent Zimmerman at that time, in patient cells, so in fibroblasts of patients. And so we actually connected this defect to a defect in calcium homeostasis, mainly a reduction in calcium content into the ER, which leads to also an ER stress response, which translates to an upregulation of the PERK, phospho-EF2 alpha pathway, leading eventually to axonal degeneration. So based on these findings, we set up two therapeutic approaches, which is one aiming at restoring this ER mitochondria interplay using gene therapy, and the other to try to modulate this ER stress pathway using pharmacological compound. So as a gene therapy approach, we try a gene supplementation strategy based on the overexpression of MFN2. So we choose mitophysin 2 overexpression, since as it is expressed both at the ER and mitochondria, we believe that it can really restore our contact. So we use AAV9 that drive MFN2 overexpression under the control of a human synapsin promoter to specifically overexpress in the affected neurons. And we first injected our mouse model at the pre-symptomatic stages. And then we assess this effect on the locomotion and histological defect. So we know that in this model, it's mild neuropathy, but we still have some readout that we can assess for therapy. And very interestingly, we were able to see that the pre-treatment was sufficient to prevent the locomotion dysfunction that we assessed thanks to the accelerating rotarod test. And we were able to connect this benefit to prevention of axonal pathology that we measure through the statin-2 levels that has been shown to be upregulated in the tibial nerve in the rat, CMT2A rat model, and that we were able to reproduce. And also, we had the rescue of the ER mitochondria contact in the motor neuron, the spinal cord of these mice. And we were really also, Happy to see that when we delivered this AAV also at the onset of the disease, so it's between 4 and 5 months, we were able to also restore the locomotion dysfunction, and this was again correlated to an improvement of normalization of the STATMIN2 levels and an improvement of ER mitochondria contact. But at the step of the study, we were not, stage of the study, sorry, we are not able really to see whether our beneficial effect was really due to an improvement only of the ER mitochondria coupling, since we also, it's not presented here, we also show an effect, of course, on the mitochondrial length. So to determine this point, we engineer, we design version of MFN1, which is the homologue of MFN2, but we were able to target it to the ER by adding a ER signal sequence. And we also inject this constriction presymptomatically. We show, I don't have time, but of course it restores the contact between these two organelles. And we were really happy to see that it also allowed to improve the locomotion function at a level similar to the overexpression of MFN2Y type, meaning that really enhancing the coupling of these two organelles is really important to rescue CMT2A. So during the meantime, we also assess whether we can have a therapeutic benefit, this time by targeting the ER stress. So we use the cefin-1 compound, thanks to a collaboration with Pierre Mignot and Inflectis Bioscience at the time, to target the PERK phospho-EF2 alpha pathway that we have shown upregulated in our model. We here use first motor neuron derived from iPS from patients carrying different mutations in MFN2, and we were able to confirm that indeed we have had an upregulation of the PERK levels and phospho-EF2 alpha in these neurons. So we started the cefin treatment, and we saw an improvement of neurite length that is completely reduced in CMT motor neurons, and also it completely prevents the upregulated of STATMIN2 levels, and even it has also a beneficial effect to restore the mitochondria length, which is an indicator of mitochondrial fusion. So based on this encouraging in vitro data, we move in vivo. We started a 6-week treatment, started 2 weeks before the symptom by delivering the drug twice a day using micropipette guided drug administration. And it provides, after 6 weeks of treatment, an improvement at the level of locomotion, and also it was correlated to a normalization of PERK pathway in the spinal cord of these mice, and this is still ongoing, but maybe also a normalization of STATMIN2 levels in the nerve, so maybe an effect also on axonal degeneration. So I hope that thanks to this data, I convince you that the targeting of ER mitochondria coupling and ER stress should be maybe considered in the therapeutical development also for CMT2A besides the targeting of mitochondria. And to finish, I would like to thank all the team members, and especially Zainab and Marine, who did this incredible job, all the collaborators, platform and fundings, and all of you for your attention, and we'll be very happy to take any question.
Did you saw changes in MFN1 levels?
You mean after the gene therapy? Yeah. Or I haven't checked this. Yes, you're right. To see if there is maybe a compensation. Yeah, it should be good to look at it.
Yes, thank you very much, Nathalie. Do you have any data regarding to the mitochondrial network, morphology, behavior, fusion and fission after the treatment?
You mean the pharmacological one or the both? Gene therapy? Yes, actually we see an increase in the length of mitochondria in the nerve, but also in the spinal cord of this mice. So it means we were able to restore also proper mitochondrial length. Yes.
Okay, thank you.